Welcome back to the Science Experience, where we explore the most exciting new research, papers, and preprints just for you. Before we dive in, please do take a moment to like this video and subscribe to our channel. It truly helps support the Science Experience and lets us bring you more fascinating science. So today we're doing a deep dive into neuroinflammation. It's a topic that's, well, surprisingly central to almost all neurological conditions. You might think inflammation is just, the body reacting to an injury. Pretty straightforward. But our sources, particularly a really comprehensive review by Fudong Shi and V. Liang, they show a much more complex picture, almost paradoxical, actually. Our mission today is to unpack this tricky immune response in the brain. We want to understand its dual nature, its different roles in diseases like MS, stroke, Alzheimer's, Parkinson's, and what it all means for future treatments.
Right. And neural inflammation, put simply, it's really just the brain's immune system at work. It means activating its own immune cells.
Yeah.
Those are mainly microglia and astrocytes. Think of them as the brain's dedicated little security guards and support crew. And sometimes, other immune cells from the rest of the body can get involved too. They can infiltrate.
Okay, the brain's own defense system.
Exactly.
Yeah.
But here's where it gets really interesting and kind of counterintuitive. While this inflammation initially tries to fight threats, maybe repair some damage, it often sort of goes rogue. It becomes like a persistent flame, fire that just keeps burning and actually starts fueling neurodegeneration.
Wow.
Yeah, it creates this vicious cycle of more injury, worsening disease.
It just feeds itself.
A fire that starts itself instead of putting one out. That's
a huge paradox.
very cool.
So if this fire is common across these different diseases, MS, Alzheimer's, etc., does it always look the same, or are there key differences?
That's a really crucial question. You see neuroinflammation as a hallmark in all of them. MS, stroke, AD, PD. That's true. But the role of those microglia we mentioned, that can be critically different.
Okay, how so?
Well, take MS, multiple sclerosis. In MS, microglia activation often seems harmful right from the start, like it's directly contributing to the damage. But, and this is the twist in Alzheimer's and Parkinson's disease, that initial microglia activity can actually be good.
Good.
They can start by clearing out harmful stuff, like those misfolded amyloid-β proteins in Alzheimer's, the sticky plaques.
Ah, okay. So they're doing their cleanup job initially.
Exactly. But the problem comes when this activation just doesn't stop. It becomes chronic. And then these helpful microglia can turn destructive.
So the brain's cleanup crew starts off great, tidying up, but if they get overworked or stuck on, they become part of the damage. It's a massive difference. What about immune cells coming in from outside the brain? You mentioned infiltration. Does the timing of that differ too?
It absolutely does. Big difference there. In conditions like MS and acute stroke right after a stroke happens, you often see a significant wave of immune cells coming in from the body quite early on.
Okay, early wave from the outside.
Right. But contrast that with Alzheimer's and Parkinson's, and also the later progressive stages of MS or chronic stroke. In those cases, the neuroinflammation seems to become much more compartmentalized. It's happening within the central nervous system, inside the brain, and spinal cord itself.
So the fight moves from being an outside invasion to more of an inside job, so to speak.
Precisely. The main action is happening behind the blood-brain barrier.
Okay, if the battleground shifts like that from outside the CNS to inside, that must completely change how we think about treating these diseases, right? I mean, what does that imply for current treatments, like maybe for MS?
Oh, it changes the therapeutic strategy fundamentally. A lot of the successful MS drugs we have now work by affecting immune cells outside the brain. They basically reduce the number of those cells getting into the CNS in the first place.
Right, stop them at the border.
Exactly. But for conditions where the inflammation is already well established inside, like progressive MS or AD or PD, well, those drugs might not be as effective. For those, you need medications that can actually get into the brain. They need to be CNS penetrant.
They have to cross that blood-brain barrier.
They have to. That barrier is designed to keep things out, which is usually good, but it's a major hurdle for drug delivery in these cases.
So we need brain-penetrating drugs. Are people actually developing those? Are there any specific targets that look promising for these compartmentalized conditions?
Yes, absolutely. There's a lot of focus here. One promising area is Bruton's tyrosine kinase inhibitors, or BTK inhibitors. These are molecules that target key signaling pathways inside immune cells, including B cells, macrophages, and importantly, those microglia within the CNS. They're in clinical trials now, especially for progressive MS.
Okay, BTK inhibitors. Anything else?
Yeah. There's also really exciting work on things like TREM2 agonists. TREM2 is a receptor found on microglia. The idea is to sort of modulate or fine-tune microglia activity. Boost the good stuff, dampen the bad stuff.
Brain tuning. I like that.
Right. Because the big challenge, the broader goal, is to develop therapies that can calm down the harmful aspects, like the neurotoxicity, without shutting down the beneficial functions, like clearing debris. It's a delicate balance.
That makes total sense. You don't want to throw the baby out with the bathwater, essentially. Stop the bad without stopping the good.
Exactly.
And given all this complexity, the different roles, the timing, the location when you actually give a treatment must be incredibly important too.
Paramount. Absolutely critical. Because the inflammatory dynamics are so different, the ideal timing for intervention is almost certainly going to vary between these conditions. For MS, hitting it early, maybe stopping those cells getting in can be key. But for Alzheimer's or Parkinson's, where microglia might be helpful early on, well, you wouldn't want to block that helpful phase, right? So the timing becomes much trickier.
It's not one size fits all.
Not at all.
So this deep dive really hammers home that neuroinflammation isn't simple. It's not just good or bad. It's this incredibly nuanced, dynamic process at the heart of so many devastating neurological diseases. And understanding these intricate differences, but also the common threads, that seems to be the absolute key for unlocking future treatments. And for you listening, it's pretty sobering, isn't it? Neurological disorders rank first globally in causing morbidity, illness, and disability. So figuring out this neuroinflammation puzzle isn't just academic, it's a critical step to reducing that huge burden worldwide.
It really is. And it leaves us with a fascinating question to think about, doesn't it? How might future therapies get really precise? Could we actually modulate these delicate immune responses? Maybe even someday sort of retrain microglia to keep doing the good stuff while stopping the destructive actions.
Retraining microglia.
It opens up some really exciting possibilities for brain health down the line.
Absolutely. Well, that's all the time we have for this deep dive. Remember to like this video and subscribe to the channel for more insights into the latest science. We really appreciate your support for the SANS science experience.